All right, so welcome everybody to one more instance of El Zoominario. Today, I have the immense pleasure of introducing our guest and my close friend, Professor Miguel Reyes. Miguel was born in Mexico City, and then he moved to the US when he was just 10 years old. He did his undergrad here in the US at the University of Connecticut. He majored in ecology and evolutionary biology. Once he completed his undergrad studies, he went on to study a master's at Southern Connecticut State University, where he also worked at the genetics lab at Yale as a researcher. Then he pursued his PhD at Clark University, and during his studies of the PhD, he focused on stress and growth using the tree spine stickleback fish populations in Alaska. And then he joined, right after finishing his PhD, he joined Emory University as a postdoctoral researcher, where he studied stress and microbiome interactions using the P-AFID model system. A little after that, he joined Emory University as a faculty member in 2020, and that's exactly when the pandemic started. And on a personal level, I can tell you that Miguel is a proficient judoka. He's also an expert scuba instructor and an occasional climber. And personally, I can't have enough of this guy, and I can't wait until we can meet again in person. So Miguel, thank you so much for being here. Welcome to El Seminario, and please take it away.
Excellent. Well, first of all, thank you, Daniel, Claudia, for such a wonderful invitation with University of Wisconsin in Madison, a great institution. And it's really an honor to be part of this collaboration. And you two are extremely close friends of mine as well. So I'm extremely happy to be here. So it is my pleasure to also thank our guests who are visiting and who joined us for our talk. So I'm just going to, for a few minutes, I'm going to be talking about my research going from my PhD and the transition that I did as a postdoc at Emory. So the title of my talk is basically, does stress help us adapt and survive? I put that as a question because really that is the question where I'm focusing most of my research. And that, of course, is my PhD and postdoctoral research at Emory. So I'm not going to talk too much about this. Daniel did a wonderful job on an introduction telling my academic history. So the one thing that I do want to say is that I've been extremely lucky, especially thinking of my PhD program on the places that I was able to visit and do my work at. Here, for example, we could see the scenery in Alaska, where I spent quite a few summers. I would take students for a month or so and do some research there, collect specimens from different bodies of water, fish populations, bring them back to our laboratory. And just to give you an idea of the places where I was very fortunate to do my work. And I just really memorable and extremely fortunate to have that experience. So without further ado, I'm going to jump really about my research as a PhD student at Clark University in Massachusetts, in the state of Massachusetts. So I do want to introduce the model system. This is the three-spine stickleback. This is a fairly small fish. I'm going to move a little thing here. This is a fairly small teleast fish, and I'm just going to give a few details about it. It is sexually dimorphic, meaning that the males and the females appear different, they look different. You could see here, for example, the coloration. Males have a very bright coloration, females tend to be very drab, gray, whitish coloration, and also the females tend to be larger, and that of course is an adaptation for carrying the eggs upon the sexual reproduction. Other things about this particular system is that it's unique in the sense that it has three different morphotypes. So, for example, there are three different major shapes that are related to this particular system in the bodies of water. One, which is oceanic, as the name suggests, is found in the ocean. And the other two tend to be freshwater, benthic and limnetic. And depending on the habitat, they're going to be somewhere within these two different morphotypes. It can be as a spectrum. It can be sort of seen as a spectrum. And I'll talk about this in the next slide. They are endemic to the northern hemisphere. So if you were to just visualize the planet, they are basically found all over the northern hemisphere, close to the shore, in all kinds of different bodies of water. And it is considered a living fossil for a reason that I will talk about now. So if we compare the ancient stickleback that lived millions of years ago with the modern ocean morphotype, they are pretty much identical to each other. So even though, we really can't say that today's oceanic is the ancestor, we could use it as a surrogate. And that becomes an important issue. And in biology, this particular system of the three-spine stickleback is considered a supermodel in evolutionary studies. So let's take a look at the different morphotypes. Here we have on the periphery of our wheel all the different freshwater morphotypes. There are certainly a lot more, but this just gives us a sample. And in the middle, we have the oceanic modern morphotype, right? Now, if we sort of look at this and associated with similar wheels, for example, Darwin's studies with finches, for example, well, we have all types of different modern finch populations, but we don't know what the ancestor looked like. Well, in the case of the stickleback, in the three-spine stickleback, we do have a very good idea of what the ancestor looked like. So by using this as a surrogate ancestor, we can then infer polarity and adaptive value in whatever traits we are studying in our particular system. So that basically gives us that added benefit of adding that surrogate ancestor. Let me just walk you through the cycle of a stickleback in any given year. And this could essentially signify a year in Alaska, right, where I spend my studies. So for example, May and June, new eggs are born. June, they're growing, everything is going well. July, October, most of the growth takes place. And of course, we're dealing with Northern Hemisphere, very high, right? So from anywhere to November to April, we have 6 months of basically freezing. We call this the over-freezing environment, where basically the surface freezes and the fish just try to survive as much as they can. And again, the ones that do survive have to reproduce somewhere between May and June. So they have a very small window of reproduction for the population, for that generation to be able to survive, right? The other concept that I want to also introduce regarding my research is this notion of compensatory growth. Now, if we sort of think about growth, right? We intake nutrients, our bodies grow, they utilize those nutrients for all kinds of things. One of them, of course, is to get large and to develop, to continue to develop and grow, right? So there are various scenarios in which, for example, we could measure weight across time and the amount of nutrients that are given over time, right? So for example, if we have a control, right? And let's say we feed our control just a regular normal diet, the control is going to follow a steady normal line. But if at some point we decrease the rations within the diet, well, the consequences are going to depend on how it's going to respond to the moment that it's fed once again. So for example, we stop or we reduce the diet. And at some point, depending on what we do, well, if we do not regain the diet, there is no compensation, there is no growth. We could, for example, have partial compensation where there is some growth achieved, full compensation where it's going to match the control, or in many cases, you could actually overcompensate. So this is basically showing us that if you starve, if an organism starves, and at some point it regains its nutrients, it may overcompensate, it may overgrow beyond the normal parameters. And we, of course, understand this as a faster than optimal growth beyond this period of diet deprivation, right? Now, we do know that maximal growth, it's rarely seen in nature, and there are many different biological reasons: stress, oxidative stress, the fact that you have to continuously seek rations within your diet, and that would not be quite beneficial. So there are certain limitations to how much compensatory growth happens within nature. But lucky for us, this is a trait that is quite measurable and repeatable, especially in fish, because fish, you may or may not know this, they are indeterminate growers, meaning that they do not stop growing. They are continuously growing And in theory, if you put a fish, any given fish, in a body of water, it could continue growing until it dies, right? It does not stop growing. So for my research question that I'm going to focus on for today, I had several, but for the sake of time, I'm going to talk about one, is does early life diet deprivation impact swimming stamina levels in subadult stickleback? Meaning, if we have a population of young stickleback that just hatched, and at different times do we deprived of the normal rations, what will be the impact on physical activity, right? Well, the first hypothesis that I tested on this with my research was that younger deprived populations will show diminished subadult swimming stamina ability beyond this sort of reproductive state. So before they're able to reproduce, they're going to show a diminished swimming stamina ability. The second hypothesis was that ecotypic morphological differences, like the morphotypes that I was talking about, benthic, limnetic, and oceanic, are going to show different variations independently of dietary effects, meaning that those differences, which are based on a genetic baseline, will also have an interaction, and they will show a certain type of effect on their swimming level. For my research, basically I have different populations, and here we have the different times of deprivation 1, 2, and 3. Controls were always fed ad lib, meaning that we would feed them as much as they could eat. And depending on which group they were deprived either in the second, third, or fourth month during their early life. And all of them, once they reached 9 months of age, they were tested. Now, how did I go about testing this? This is where my students in our PhD program, they were essential. I designed on paper, and I built this tank that acted as a water flume, basically subjecting the fish to different flows. And I would test how well they can swim against the flow. And in order to do this, it took a lot of time. It took about a year of work to basically build this and test it and make sure that it was accurate. I'm going to show you, by the way, I utilized almost 2,000 samples across 18 populations in Alaska. They were all independent of each other, isolated from each other. So this took years and years of work, of massive data taking with my work, and of course with the assistance of my students. So this is basically what it looked like. And I think you're going to enjoy this. Every time I give this talk, this is people's favorite part of the talk, when I show what the test looked like. So here we have this little fish, right? Before I did the actual test, I would acclimate it to a certain slow flow. You could see here, it's a very gentle, not particularly swimming hard. I did this for a couple of minutes just to acclimate it. And then I would jump the rate of the flow to 18 bodies per second. Basically, that's the equivalent of a human jumping and just swimming as fast as it could. And I would record how long the fish can actually withstand this flow. So here we see now we have a much stronger flow. And basically I was counting in a stopwatch how long the fish could, fight off this particular flow against his body, right? And the moment that they were pushed behind the mesh and they could no longer swim, I would stop the flow. I would let the fish recover. I would take them out and put them back in its population. And I'm glad to say that none of my fish died. They all recovered. You could say that this was a rather cruel experiment, but they were in no way harmed. They were just tired. And you could see that they were breathing very fast after each given test. So for the overall results, here we have some data that I was able to publish. Before I actually defended my PhD, I was able to infer different levels of deprivation at different times and how they compare to one another, basically showing that, not surprisingly, the controls that were always fed normally, they performed the best. However, if we look at the error bars, there weren't many differences except in the second deprivation group. They were certainly much lower than the rest. So there was a little bit of age-dependent swimming stamina differences, and there were also substantial differences across the different morphotypes, right? So let's remember, you have the oceanic, which is the largest, very strong fish living in the oceans, always dealing with oceanic currents, benthic and limnetics, freshwater, not particularly dealing with a lot of flow. So oceanics overtook the other two. And if you compare the freshwater types in every single instance with some degree of error, the limnetics did overtake the benthics. And here we have a nice representation. And when I did this, we were having one of the Olympics. So as a little, pun, I actually gave them their little Olympic medals to show you the main difference in swimming capacity within them. So again, this was one piece of data that I was extremely proud of, a lot of work, and I was able to publish and show that early life changes in diet does have a substantial effect on how well you're going to act and how much stamina your body can accrue over time. So effectiveness is about all growth. We can certainly compare this and put it within the context of the life cycle, right? So it is beneficial to have a larger body size before entering overwintering, right? There is some ecotypic level variation if we sort of consider the fact that they're growing, but they have different stamina levels. And there is indeed a low dietary deficit effect. So it's better to grow, right? To just forego the dietary deficit and just try to accrue as much energy as possible for surviving the overwintering period. So this is my little sort of 2 cents on my PhD work. So now I'm going to jump into what I did at Emory University when I was a postdoc, starting 2016, and I did that for 2 years. So I'll talk about my research at Emory, and you're going to sort of wonder, well, how different is it? Well, I certainly switched models. I no longer focused on fish. I jumped into insects, which was quite daring. I did not really know much about insects, but you will notice that the types of questions that I'm asking are quite similar to the ones that I was asking during my PhD program with the fish. So I'm going to introduce to you a very different theme. We're going to talk about, for a few minutes, on the microbiome. What is the microbiome within organisms, certainly within humans, right? Well, the microbiome, which is the overall focus of my research at Emory, well, you could define it as our last, mostly unexplored organ. There is still a lot that we don't know about how the microbiome works. It consists mostly of beneficial symbiotic bacteria. These are bacteria that thrive within our body. There is some benefit that we get from us, and they get a benefit by being harbored and protected within our bodies, right? They facilitate all kinds of things within us. So for example, the ability to digest certain types of food, immune system regulation, vitamin production, and so forth, protection against pathogens, right? So all these are really important benefits that we get from symbionts. So how do we study the microbiome? Well, let me introduce to you my model system at Emory, the little pea aphid, which is a tiny little bug, which you could normally see below some of your plants. They tend to be, they are considered a pest in many different regions, but it is indeed a very important model system within molecular biology and physiology as well. So here we have the aphid. Now we can think about the kinds of things that impact the aphid, right? So for example, heat, predation, disease, not that different from any other organism. So let's talk about, this being environmental stressors and how this little insect can defend against the different stressors. Well, there is behavior. If you have predation or if you are in an unhealthy plant, well, you could get away from the plant, go somewhere else, right? You could move. It does have a rudimentary immune system, which protects it from certain illnesses. It does contain symbionts, right? You have this beneficial set of bacteria, which will protect it against certain things. And another one, of course, is the phenotype, right? If you think about the cycle of the aphid and you have a female aphid, right? When it reproduces, and something unique about this particular system is that in the summer months, it's parthenogenic in the sense that they're all females and it's essentially cloning themselves. All the females are cloning themselves. So depending on whether are stressors or not, it's able to produce either winged offspring or wingless offspring, which is a unique trait because they are genetically identical, but morphologically different. So these are the kinds of different mechanisms that it can protect against such environmental stressors. The two that I'm going to focus on are, of course, the symbionts and the phenotype. So let's talk a little bit for a minute or two about the history of the association within symbionts within the aphids. So aphids, here you can see on the left, a mother aphid, female aphid, cloning herself with all these tiny little babies that will essentially grow, and in the future, they will clone themselves once again, right? But we do understand that aphids have an obligate symbiotic bacteria. It's called Buchnera aphidicola, and this is one of the most ancient associations that we know of in the animal kingdom. Extremely, extremely ancient. Perhaps the most ancient symbiotic association between two different organisms. And what do I mean by obligate? Well, it can no longer survive without this particular bacteria. It cannot survive without Buchnera, right? If you remove the Buchnera, the aphid will most likely die, and the bacteria itself will also die without being in the aphid host. This is how we define an obligate symbiont. There are also facultative symbionts, right? Where you have certain bacteria that you could have, but you don't particularly need. They do give you benefits, but they are not required. It's not required for the aphid to have, for example, this particular bacteria, Regiela. If it has it, it may confer some benefits, but it doesn't need it for survival. So my research focused on looking at this facultative specimen, this symbiont, Regiela, and its potential protectiveness under different stressors. One of my colleagues and great mentors, Ben Parker, he was instrumental in teaching me this particular system and understanding the ability of Regiela to protect the aphid against, for example, a pathogen, a Pandora neophadis, which is a fungus pathogen, which essentially turns the aphid into a mummy. It will mummify and spirulate and send all the spores throughout the environment. We could see here, which is important to notice, is that the bacteria itself has different strains, right? And the host can also have different strains. The aphid host can have different strains. So depending on the host by bacteria interaction, you're going to end up with high levels of protectiveness or 0% of protectiveness. So this shows a nice spectrum of this interaction between the host, the aphid host, and the bacteria. For my years at Emory, when I was doing my research, I was able to get a few nice publications out of this. One of them was to look at the effect of survival when it comes to this fungal spirulation. One of my great colleagues, and he was a PhD student, Wen Hao, him and I, we were able to publish a set of experiments that was looking at the difference between infection and not infected populations and the protection against spirulation in this particular fungus. Here we have a survival plot where we could see the different lines of survival based on whether you are infected, but also its interaction on the different strain of the Regiela. So that shows a nice host by bacterial interaction as well. And the question that I was really focusing for my particular set of research points before I ended my tenure as a postdoc was to ask the following question. Do symbiotic bacteria influence this wing polyphonism in P. aphids exposed to crowding stress, right? So is there a sort of an impact of having this bacteria when it comes to polyphonism and how that relates to stress? Well, the first hypothesis was environmental stressors interact with the symbionts, and it will shift fecundity, basically how many offspring you are making, and the rate of wing polyphonism. The second hypothesis was the level of symbiotic interactions will be associated with this host symbiotic strain interaction that I've been talking about for the last few minutes. For this particular question, again, we are no longer in fish. I cannot have them swim, right? But it's a different type of stress. There is something called the overcrowding stress. And this is where I tell my students, imagine being in a room with 100 people and being stuck there for a week. I'm sure everybody would feel stressed, right? Well, the same thing happens with the insects. You stress them, you put them all clumped together in a small space, they are going to become stressed. So we have a stress group, a control group, where you basically grow them in plants without this stressing factor, right? You're going to test polyphenism, how many of the offsprings are going to be winged versus wingless, right? And what I did, which was unique in my experiment, I went to the second generation beyond the parentals to see how the impact of being wingless or winged, what type of impact that has on the proportions of the F3. So that was, in essence, one of the first times that anybody has ever done this, utilizing this particular model system. And just to show you a little bit of my data, it can be a little bit complex, but I'll try to break it down, is that we were for the very first time able to show that influence of symbionts can actually skip generations. You can have a grandmaternal stress factor shown as we did in our experiment. In this particular piece of data, I'm showing here the impact of the stress, the crowding stress, and also the impact of the maternal phenotype. And as we can see here, we have the different types of Regiella, right? And of course, we also have another group without the symbionts to see whether there were any differences or not. And in a nutshell, these came out as a very nice publication with a bunch of different authors. And I have to say, I was the only one that was not an expert on aphids. And I would have to say that this particular paper I am most proud of because it essentially made my career as an insect biologist. So I was able to show, among other things, that this particular stress is highly dependent on the interaction between the host and the type of symbiont that the host is carrying. So we could see here a nice variation in fecundity, depending, of course, on whether you're stressed or winged, and also in the amount of babies that are created in the next generation that are either winged or wingless. So you could see a nice sort of interaction between the host and the bacteria. Anyway, so let's move on for now. I was also asked very generously to talk about something that I found to be quite interesting, to relate my research and my experience as a biologist with my own culture and heritage. When Claudia and Daniel approached me with this, they told me, well, talk about something unique about perhaps your culture, your heritage. And I thought about it. And I found something that is quite fun and unique, and I hope you all enjoy it. It's not something that I have worked on before, but it relates very nicely to the types of questions that I tend to ask in my research. So for this particular piece of information, for the last few minutes before we end, I'm going to talk about this particular amphibian. You may or may not have seen it. It's called an axolotl, right? We have a nice picture of an axolotl here, the Ambystoma mexicanum species. It's called either an axolotl or in modern Spanish, ajolote, right? This is a very peculiar little amphibian that is quite popular in Mexican folklore. And I'll go through why it's such an important animal and how it relates to my own interests in research. So a brief introduction about the axolotl. Here we have a nice little representation. It is quite important in Aztec culture. Here we have a nice fresco, ancient fresco showing one of the myths of one of the gods trying to hunt this axolotl. But why is it such an important icon? Well, if you think about the Aztec language, Nahuatl, it means water dog, right? There is an association with Xolotl, which was the god of the underworld, being symbolized by a dog. And this was one of the highly linked sort of animals with this god. So it was known as a water dog. They are endemic to a very ancient lake, which some remnants of this lake still exist as channels in modern Mexico City. And if you go to these channels today in Xochimilco, you can still find populations of this particular axolotl, right? And if you think about the way that the ancient capital Tenochtitlan was founded, it was basically founded on a water basin, right? And it was full of lakes, full of channels, rather. And some of these channels still exist, right? And it was, as we understand from historical data, that it was used as food throughout the Aztec populations. And now if we switch to the biology and how I relate it to my research, it's involved in something called metamorphosis, right? It is a metamorphic salamander. And I'll explain that I'll explain what that means in the next slide. It is highly related to the tiger salamander. So they're basically cousins, right? It has really peculiar abilities to regrow limbs. And because of this peculiar ability, is a very important model in genetics and embryology in science. Unfortunately, like many other species, it is critically in danger, especially since these lakes are, highly polluted, although there are a lot of efforts to protect this particular important species. So what is paramorphosis? And now that I've been, I told you that word, so let's go through what that means, right? So if we take the cycle of any given amphibian, these are animals that are required to be close to a water source, right? You could see, for example, you go from the egg to an embryo, right, to go to a tadpole, and then you go through a particular change where you're going to lose these gills, you're going to grow lungs, you're going to lose the fin, and you grow a tail. This is basically how we can associate typical amphibian metamorphosis. But in the axolotl, curiously enough, it does not go through metamorphosis. You go from a very young tadpole all the way to adult, and you retain, the adult retains these sort of juvenile gills that are usually lost during metamorphosis. So if we compare, let's say here we have a juvenile axolotl, right, a juvenile amphibian, an adult tiger salamander, you could see that it lost its gills, its head gills, and it grew a normal tail, while the axolotl, it grows, but it does not lose its juvenile traits of gills. So paramorphosis is basically this retention of a juvenile trait, right? So axolotls do not undergo metamorphosis. What is the importance of this particular phenomenon is that they're highly adapted by retaining these gills. They are highly adapted to stress. Now, if you think about the lakes and the channels where they live, where they thrive, right? They do grow a secondary lung, but they retain their gills. Why? Because they require that added trait to retain all the oxygen that they can. If you think about the stability of these habitats, they do not have a lot of oxygen, right? So they do utilize the lungs and they retain those gills because they live in highly anoxic habitats. So this, my friends, is how I relate this particular species to my own interest. There is a very interesting trait here, which is an adaptation to a highly stressful environment, right? So, and another really interesting thing about this ancient Aztec animal is that it's 1,000 times more resistant to cancer than mammals. So again, that is another reason why this is a very important system within genetics, as well as in biology. We want to understand what about it makes it so resistant to cancer. A very, very peculiar little animal. I did mention that even though they are in danger, there is a lot of work, as we could see here in the bottom picture. There is a lot of work within the biologists in Mexico to protect these species. It's also a quite common pet that people can easily get. I never had them. I never actually dealt with them. When I was at Yale decades ago, there was a population that I was taking care of as one of the veterinary technologists. So I have seen them, but I have never directly worked with them. I would like to at some point. I think they're just fascinating. But what I was going to get to is that there is a lot of work in order to protect them and to rescue them from becoming extinct, which I think is a very, very important thing. And I will just end by showing you, a piece of really peculiar data that recently, and you could just sort of see the importance of this species within Mexico, right? Recently, there was a major change in the currency, and some of the new bills actually have the axolotl within them, which I think they're just beautiful. And I even asked my sister, she lives there, to save me all the bills that she can find. In the middle here, we have one of the books that I was given as a gift some years ago by my father. And this basically tells you the fabulous story, the myth of the axolotl, all the different stories in Aztec mythology, all kinds of art relating to this particular little animal. One of my favorite books, sometimes I'll just look at it and I'll enjoy it. Gives us a lot of really interesting information and history of this little animal. And I do want to end, and this is my very last point of information, that one of my favorite stories when we're talking about literature comes from Julio Cortázar, who perhaps is my favorite author of all. And he happens to have a short story based on this particular animal. And the story is called Axolotl. So as a little surprise, I'm going to stop sharing because I do have a little surprise that I want to share with you all. I do have PDFs of this particular story that I do want to share. So what I'm going to do is I'm going to, on the chat, I'm going to just very quickly put copies of the PDF in both English and Spanish for your enjoyment. Perhaps you have read it before. If you have not, I'm very happy to share this with you. I'm putting a Spanish version, which is the original version, as well as the English version that you can enjoy. And I'm putting that in the chat if this allows me. And then from there, we can stop. Danielle is not letting me add anything on the chat.
Thank you very much, Miguel. So I don't know if there is some issue with the chat. It seems here that you should be able to comment in there. But what we can do is I will stop sharing.
Yeah.
I will stop recording, sorry.
Sure.
And then we can, maybe it's because of that. So
yeah,
let's do that.
Okay.
Thanks, Miguel, again. Before we leave, thank you very much for such an amazing talk. And
thank you.
As soon as we stop recording this, we will start getting some questions from the audience and also take a look at those PDFs that you're going to share.
Sure, absolutely. Thank you.